In this video, we are going to conclude the topics for chapter 14. Currently, in this year, we are living in a pandemic that has actually been provoked by a virus. It all started in 2020 when worldwide Covid19, in other words, one of the members from the coronavirus family spread, and it caused many people to be infected, and worldwide a lot of people have also died. So let's actually discuss what is a coronavirus, because I want you to understand that when it comes to the coronavirus, this is not a new virus. Coronaviruses are a large family of viruses that cause illnesses ranging from the common cold to severe diseases like pneumonia and even MERS and SARS. The common symptoms of the coronavirus, including COVID-19, are fever, a dry cough, mild breathing difficulties. They can also be severe ones, as we're going to see in a few moments, gastrointestinal issues, diarrhea, and general body aches. Now, some of the more severe symptoms of COVID-19 include high fever of 100.4 degrees Fahrenheit or higher, pneumonia, kidney failure, and even death. Now, when it comes to how COVID-19 and other coronaviruses are transmitted, so far it is understood that it is through coughs or sneezes from an infected person, or if an individual has touched a contaminated object. Now we are going to explore how COVID-19 attaches to cells and how it is replicated. As you can see, we have here a figure of what is understood so far of the coronavirus, including COVID-19. As you can see, coronaviruses in general, and I'm just going to highlight the important information, bind the cell surface, of their host cells. It is understood that COVID-19, one of its major targets when it comes to identifying the cell that is going to be infecting for its replication is pulmonary cells. That's why some of the severe symptoms discussed in the previous slide are actually observed. Now, as you can see, some of these cell surface molecules that are in the host cell are going to include metalloproteases, aminopeptidases, so on and so forth. Now, understand that there's also a co-receptor that it is known to be an N-acetyl neuraminic acid, So, it is not understood, it is not clear yet whether the virus get into the host cell by fusion of the virus and the cell membrane or by receptor-mediated endocytosis, which means that in endocytosis, this virus is actually engulfed by the cell membrane, and that's how it enters the cell. So it's not well understood. Now, understand that when it comes to COVID-19, it is known as an RNA virus. In there, the coronavirus, including COVID-19, have a single positive stranded RNA genome. When we have a positive single-stranded RNA genome, that means that it is called positive because the strand runs in the 5' to 3' direction. Now, at first, it is known that the virus synthesizes, its RNA polymerase molecule that only recognizes this single positive RNA stranded molecule And what it does is that viral RNA polymerase is going to synthesize the minus strand, utilizing the positive strand as a template. So what that means is that it's going to synthesize an RNA molecule in the 3' to 5' direction. After that negative strand has been produced, understand that it serves as the template to transcribe smaller genomic positive RNA that are used to synthesize all the other proteins that are needed for the virus to be regenerated. Okay? Understand that this negative strand also serves for the replication of new positive stranded RNA genomes for when the virus is replicating. Now, understand that there's other important proteins that are going to be obtained from this genomic RNA that comes from the virus. Those include protein N, protein M, which, as you can see, they are actually integrated into the membrane and the endoplasmic reticulum. So in essence, what is happening is that after binding, these assembled nucleocapsids with helical twisted RNA buds enter the ER, the endoplasmic reticulum of the host cell, and they are going to be encased within its membrane. And we can see that specifically in this area of our diagram. Understand that in the final step of the replication of the coronavirus, the resulting viral progeny, okay, is transported by Golgi vesicles into the cell membrane, and they are going to be exiting the cell into the extracellular space, which means that that is outside of the cell. So as you can see, this process, it is complex, but the main point that I want you guys to understand is that when it comes to COVID-19, this is not a retrovirus. Here, what we have is a virus that has, as part of its nucleic acid RNA, but because it contains a viral RNA polymerase, it actually makes more RNA, and then from that RNA molecule gets replicated to make more RNA molecules, and those RNA molecules then can actually be translated to make the proteins necessary to make the virus particles that can exit the host cell. Now, currently, one of the main recommendations that have been given by doctors and even the health association around the world is to wash your hands as you move in everyday life and even when you come home. So you may be wondering, why is it that soap will work against specifically viruses or in addition to the COVID-19 virus? One of the things is that it is known that the coronavirus has a membrane that it is oily, meaning that because it has this lipid membrane that contains the spikes, okay, when soap molecules interact with it, remember, a soap molecule is going to have a hydrophilic head and a hydrophobic tail. In essence, what happens is that this lipid membrane, which it has components that are non-polar, the soap is going to destroy the virus by the non-polar tails, which I'm highlighting in pink, are going to now interact with the non-polar portion of our lipid membrane. And overall, since they wedge themselves into the lipid membrane, they are going to open it and pull it apart. Okay. Once that lipid membrane has been pulled apart, as you can see, the spike proteins and sections of that pulled apart membrane are going to be trapped by the nonpolar tails of the soap. We are going to create a micelle, and a micelle is actually a molecule that it is soluble in water, so it can be washed away the components of our virus. There are other methods that are utilized in order to also have effects for the COVID-19 virus. Okay. One of the other target methods for COVID-19 is through small molecules. Okay. So one of the treatments for COVID-19 is the treatment with Remdesivir. Okay. Remdesivir, it is actually a molecule that it is considered a broad spectrum antiviral treatment that was first developed by to treat Ebola. Now, when it comes to structurally how Remdesivir, which you can see the molecule on the right side of the slide, the reason why it can actually work against COVID-19 is because Remdesivir mimics one of the building blocks in the virus genome. In addition, it can interfere with the RNA polymerase enzyme that we discussed previously when we went over the replication of COVID-19 and other coronaviruses. So it can actually interfere with that RNA polymerase enzymes, which is what, as we saw previously, replicate the genetic material from the virus. So overall, since it is interfering with the RNA polymerase enzyme, it can act against the virus replication. Okay, so in this example specifically, as you can see, COVID-19 is also known as SARS-CoV-2. So understand that this is also talking about COVID-19. Lastly, in more recent times, vaccines have also been developed as a treatment for COVID-19. The current technology that it is utilized for the COVID-19, understand that it is an mRNA vaccine. This mRNA vaccine has been developed to produce the following. So an mRNA vaccine is basically a vaccine which is made from the mRNA, which basically it gives the instructions to host cells for making viral proteins. Now, this viral protein that it is packaged in lipid nanoparticles are delivered to the patients via injection. And the idea of an mRNA vaccine is that this mRNA is released into the cell. Inside of the cell, this mRNA is going to be converted by the host cell to make viral proteins. And at this point, many of the vaccines that are commercially available are making the spike protein in COVID-19. And the intent is that once these spike proteins that came from that mRNA, as you can see here that I'm highlighting in purple, then the immune system is going to create antibodies for identifying specifically those spike proteins, and that it is suggested that it is one of the ways in which we can create specific antibodies against identifying the spike protein specifically of COVID-19.